The overall goal of this procedure is to quantify the chemosensitivity of multiple myeloma primary cells to a panel of chemotherapeutic agents. This is accomplished by first mixing multiple myeloma cells isolated from fresh bone marrow aspirates with pre-culture patient-derived stroma and collagen. In the second step, the cell mix is seeded onto a multi-well plate. The cells are then treated with the chemotherapeutic agents of interest, and the plate is transferred to the incubator of a motorized stage microscope for sequential imaging of the cells over a 96-hour incubation period. Ultimately, a digital image analysis algorithm can be used to generate viability curves for each drug and concentration.
The main advantages of this technique over existing endpoint viability assays are that it can be used to test several chemotherapeutic agents versus a single patient specimen, and that the viability of the patient cells can be measured longitudinally over several days without needing to separate the cells from the stroma or the matrix.
The implications of this technique extend toward the therapy of hematopoietic tumors, as it can theoretically be used to predict clinical response to specific chemotherapy regimens.
We first had the idea for this protocol when we realized that replacing our original microfluidic slides with multi-well plates would greatly reduce the cost and time required to complete the experiment.
To prepare primary multiple myeloma bone marrow-derived mesenchymal stem cell co-cultures with a multi-channel pipetter, begin by suspending the cells at 6 times the final concentration in rPMI 1640 medium. Next, mix 50 microliters of each cell type with freshly prepared type 1 collagen to a final